Hello, dear students. We will start the next lecture, lecture number 22 of Physiology U301. We are studying since last lecture that reproduction and endocrine glands. So during reproduction, we have seen that how the sperm are formed, or we have seen the spermatogenesis. In spermatogenesis, we have seen two phases, and among these two, from the first page, there are also three sub pages. That is, formation of spermatid is the first page, and phase of multiplication, phase of growth, and now phase of maturation. Because the cells which are multiplied in first, the phase in which they are grown well, and in the phase 3, how they get mature or how they get developed for the production in the next stage. Is because next stage, second stage, is spermiogenesis. That we will see. The in phase of multiplication, they are multiplied in their numbers, and in maturation they got matured completely, developed. The primary spermatocyte becomes secondary spermatocyte, and then it also develops and matures, and from n number of chromosomes, each secondary spermatocyte have single n or haploid number of chromosomes, and each secondary spermatocyte undergoes second meiotic division or second maturation division, just like that of mitosis, and produce two spermatids, and which is haploid n number. Therefore, we can say 2n number, n and n. Thus, at the end of meiotic division, a diploid spermatogonium, 2n number, produces 4 spermatids, and which are non-functional male gametes, to become functional spermatozoa, and they have to undergo a process of differentiation of specialization, the spermiogenesis. The second process is spermiogenesis. Spermiogenesis means differentiation of spermatids into the sperm is known as spermiogenesis. Differentiation means they might have different structures. The sperm is very active, motile cell, and this motility is provided by discarding superfluous material of developing sperm, or we can say spermatid. And the n number of spermatids are formed, and that is a typical cell that containing nucleus and cytoplasm. The cytoplasm contains the cell organelles like mitochondria, centrioles, Golgi bodies, and two major parts of the spermatozoon are the head and the tail. The head is globular, somewhat ellipsoid. Anteriorly, there is a proteinous material called as acrosome, and the tail, which is very thin, but in between tail and head there is a middle piece, or to which we can call it as a principal piece, and the end piece. And this is the differentiation, or we can say the spermiogenesis is completed here. Now, formation of head in spermiogenesis, or formation of sperm. What is there? Head and tail. So in between head and tail we have seen middle piece where mitochondria are arranged there. And how the head is formed, formation of head. The two major parts of sperm, that is head, the nucleus and acrosome. The nucleus is at the center of the head and acrosome is at the tip, undergoes the following changes to form the sperm head. So changes in the nucleus, the changes in nucleus occurs. What changes? The nucleus of spermatids shrinks and losing much of water from the nuclear sap, and assumes different shapes in different animals. Therefore, the structure of sperm is different in different animals. The DNA becomes more concentrated, and the chromatin material becomes closely packed RNA, most of its proteins, and nucleolus are greatly reduced. Second, in further nuclear changes we have seen, then in second, acrosome formation. At the head of the sperm, there is a dark blackish point where the protein material is aggregated and which helps to dissolve the wall of ovum from where the sperm is to enter and the fertilization carried out. That is nothing but the acrosome. How it is formed? The acrosome of spermatozoon is derived from the Golgi bodies of the spermatid, and during acrosome formation, one or more vacuoles of Golgi bodies, or we can say Golgi complex, starts enlarging and occupies the place between the tubules of Golgi bodies, and that helps to aggregate these molecules to the anterior side, and after gathering it forms a blackish part. We call it as the acrosome. But how? Soon after, a dense granule known as the proacrosomal granules develop inside the vacuole, and the proacrosomal granule attaches with the anterior end of the nucleus and enlarges into the acrosome. The membranes of Golgi vacuoles form a double membrane sheath around the acrosome and form a cap-like structure of the spermatozoa. The rest of the Golgi bodies become reduced and discarded from the sperm as Golgi rest. Means whatever the Golgi body, whatever the protein molecules of the Golgi bodies and the vacuoles from the Golgi bodies, which are prepared in between these Golgi strands, are aggregated at the anterior side and forms a moon-like structure at the anterior head of the spermatozoa, and that is called as acrosome, which helps to dissolve the wall of the ovum for the entry of that sperm in that ovum, and fertilization occurs. And whatever the material rest after formation of this acrosome, this is called as Golgi rest. So second, formation of tail. In first, the formation of head, formation of acrosome, and second, formation of tail. So in that, first the role is of centriole, because the two centrioles of the spermatids become arranged one after the other just behind the nucleus, and the anterior one is known as the proximal centriole and the posterior one is known as the distal centriole. So centrioles are arranged one and the other poles, anterior and posterior pole of the nucleus, and therefore they are proximal and distal centriole. The distal centriole changes into the basal bodies and gives rise to the axial filament of the sperm, and in this way the actual strand of the tail is formed here, and the distal centriole and the basal part of the axial filament present in the middle piece of the spermatozoa tail, and in this way the centriole forms by enlargement of the material at the posterior end and forms finally a tail. Second, the mitochondria. As usual, we are well known about the mitochondria because these are the energy rich compounds, or we can say energy generating energy, or we can say calorie, or we can say powerhouse of the cell in simple way. Most of the mitochondria concentrate around the proximal part of the axial filament to form the middle piece of the spermatozoon tail, and the tail of sperm comprises an axial filament which remains differentiated into principal piece and end piece. So these are the two pieces of the tail. Third part, changes in cytoplasm. Since nucleus we are observing all the changes, now at last What are the changes takes place in cytoplasm? So just prior to the termination of spermiogenesis, most of the cytoplasm is discarded from the sperm, but the plasma membrane forms an envelope around the head and middle piece and main piece of the tail. So plasma membrane plays a key role to envelop around this material as a protection. The end piece of the tail remained naked. It has no envelope and hence consists only terminal part of the axial filament. Means there is no encasement or no envelope of the plasma membrane. In this way, all the changes occur in the formation of sperm, or we can say spermiogenesis. Now, hormonal control of spermatogenesis. How the spermatogenesis occurs? With the help of hormones, because hormones are the chemical messengers which carry the messages to perform that particular act either in the testes or ovary or any other organ in the body. So hormones convey the messages or signals. What are these? We will see one by one the name of the hormone and their roles. So there are some hormones and their roles. First is the testosterone hormone. Testosterone is secreted by the Leydig cells located in the interstitium of the testes and is essential for the growth and division of the testicular germinal cells, which is the first stage in the forming sperm. So testosterone is the hormone, male hormone, secreted by the Leydig cells, or we generally called as interstitial cells, which are close to the interstitial cells, and they are needed for the growth and division of the germinal cells because they grow the germinal cells. Second is luteinizing hormones, or we can call it as the LH. These hormones are secreted by the anterior pituitary gland. Pituitary gland is the master gland among the endocrine glands, and its secretion is very important. Luteinizing hormone that stimulates the Leydig cells, these Leydig cells, to secrete the testosterone hormone. Means this hormone is formed in these cells, and this is essential for the growth, and it is secreted or it is stimulated for secretion by luteinizing hormone, another hormone. The formation of one hormone is depending on the stimulation of another hormone. This is one important thing in the endocrinology here, and which in turn stimulates the process of spermatogenesis. Means these both together perform the activity and the actual spermatogenesis process is started here. Third one is the FSH, we call it as follicle stimulating hormone. This hormone is secreted by the anterior pituitary gland and stimulates the Sertoli cells. We have seen the Sertoli cells and spermatogonial cells in the seminiferous tubules in the testes. So these cells are stimulated, Sertoli cells are stimulated by this hormone. Without this stimulation, the conversion of the spermatids to the sperm will not occur. Means the process of spermiogenesis or the formation of sperm is actually stimulated by this hormone, and then the activity of spermatogenesis is facilitated. Fourth hormone is estrogen. It forms from testosterone by the Sertoli cells when they are stimulated by the follicle stimulating hormone, are essential for spermiogenesis. So estrogen hormones stimulate the spermiogenesis, and it is formed by the Sertoli cells which are present in the seminiferous tubules. Because spermatogenic cells or generative cells which form the sperm are supported by four Sertoli cells at the four sides, and they secrete the hormone estrogen. and they are essential, very necessary for the spermiogenesis. Means we have seen the differentiation of sperm. So in this way, the spermiogenesis activity is completed here, and next to that is the process of development of sperm. And then, according to the syllabus, we will go to the next topic of hormone and its role, that we will see in the next lecture. Till now, we have this a brief lecture here. So see you for the next lecture.